Imagine this. At the very edge of Europe, where the North Sea beats against the low shores of the Netherlands, lives a people whose genetic code holds a mystery that is 1,500 years old. In 2002, geneticists took DNA samples from 94 Frisian men and compared them with residents of central England. The results shocked even the scientists themselves. Two peoples, separated by the sea and centuries of different histories, turned out to be statistically indistinguishable in their male genetic lines. As if the North Sea did not exist. As if someone had transported the genetic code of an entire nation to an island. Who are these people? Why does haplogroup R1BU106, the main marker of North Sea Germanic peoples, connect them to the English more strongly than any history textbook? And what is hidden behind the phrase, the last Anglo-Saxons of Europe? The answers lie in the DNA molecules, and they will surprise you. Today, we will examine 4 key points. First, why the Frisian language is closer to English than any other in Europe, and what this means from a genetic perspective. Second, what the DNA analysis of more than 2,000 men from the Netherlands revealed, and which haplogroups dominate on the North Sea coast. Third, how ancient DNA from Anglo-Saxon burials from the 5th to 8th centuries pointed to the Frisian coast as one of the main sources of the Great Migration of peoples to Britain. And finally, why modern Frisians have preserved in their genes what has long dissolved in the flow of later migrations among the English. And at the very end, I will tell you about a discovery made from the analysis of 460 ancient genomes that compelled scientists to entirely reconsider the scale of the Anglo-Saxon migration to Britain. But let's go through everything step by step. In recent years, DNA sequencing technologies have allowed scientists to look into the past with a precision that was once only a dream. What had been a subject of dispute among historians and linguists for centuries is now being verified down to the molecules. In 2022, an international team of geneticists published the results of the largest study of ancient DNA from the Anglo-Saxon era in the journal Nature. 460 ancient genomes, of which 278 were from burials in England, dating from the 1st to the 13th century. These data overturned previous understandings of who actually contributed to the English gene pool. It turned out that the answer must be sought not on the British Isles, but on the other side of the North Sea. There, where the flat polders stretch to the horizon, where the wind smells of salt, and where people still live whose DNA holds the key to one of the greatest migrations in European history. Who are these mysterious Frisians? Did you know that linguists identify a special subgroup of West Germanic languages called Anglo-Frisian? Old Frisian and Old English form the closest pair within it. These two languages share unique phonetic and grammatical features that set them apart from all other Germanic languages. Such closeness signifies that at one time, both languages were one and the same. Historically, the Frisians occupied an elongated strip of the North Sea coast, from the Dutch province of Friesland through East Frisia in northwestern Germany to North Frisia in Schleswig-Holstein. In the early Middle Ages, their territory was even larger, encompassing a significant part of the coastal lowlands. The linguistic proximity itself indicates a common origin of the populations on both sides of the sea. But linguistics is a hypothesis. What if it could be tested? That's exactly what geneticists did. In 2013, a group of scientists led by Abdellaoui conducted a large-scale study of the genetic structure of the Netherlands. They analyzed the DNA of 999 men from 54 locations across the country. 443,816 genetic markers for each. At first glance, the results seemed dull. The average genetic differentiation between subpopulations was just 0.000038. Almost zero. It seemed that all Dutch people were genetically identical. But when the scientists looked closer, an invisible boundary emerged. A clear microgeographic gradient from southeast to northwest. From the southern provinces to the North Sea coast, the genetic portrait of the population changed smoothly. Even within one small country, DNA paints lines that are backed by millennia of history. And the most interesting discoveries were hidden in the north, where the Frisians live. But the real breakthrough occurred in 2020. Byrne and co-authors applied the haplotyping method to the data from 1,626 Dutch individuals. This method compares not individual markers, but entire segments of chromosomes, which provides a much finer picture. The researchers discovered 40 genetic clusters across the country. When aggregated into 16 groups, one cluster attracted particular attention. They designated it with 4 letters: NHFG. Northern Holland, Friesland, Groningen. 3 provinces on the North Sea coast. This cluster differed from the rest of the Netherlands in terms of lower effective population size, increased relatedness among individuals, and signs of a founder effect. Simply put, the residents of the northern coast turned out to be an older population, preserving a genetic portrait that had long changed in the southern part of the country. Friesland began to stand out from the general picture, but that was just the beginning. By the way, a fun historical fact. When scientists applied the Globetrotter algorithm to the data from the northern cluster, they discovered something unexpected. In the genes of the residents of these 3 provinces, they recorded a mixing event with a significant Danish component. The algorithm dated it to the interval from 759 to 1290 AD. This is precisely the period when historical sources document the presence and rule of Danish Vikings in the northern territories of the Netherlands. Genetics confirmed. They didn't just come. They left their mark on the DNA of the local population. But here's what's important. Despite this Danish layer, the Frisians preserved their ancient genetic foundation. The Vikings added a new hue but did not repaint the picture. In modeling external contributions for the northern provinces, the share of the Danish component was found to be higher than in the south, but the foundation remained the same. And now, let's shift from the overall picture to the male lines. That's where the main clue lies. And this brings us to the most intriguing part. Ready for shocking statistics? In 2020, Altina and co-authors published the most detailed study of male genetic lines in the Netherlands to date. 2,085 men, 99 collection points across the country, 92 markers on the Y-chromosome that allow differentiation of 32 haplogroups. The Y-chromosome is strictly passed down from father to son and changes very little over generations. This makes it an ideal tool for tracking male migrations deep into history. And here is what the analysis showed. 2 macro haplogroups absolutely dominate. Group R, predominantly R1b, comprises almost 62% of all male lines in the country. Group 1 constitutes 27.82%. Together, they account for almost 90% of the male gene pool of the Netherlands. And the remaining 10% is shared by all other lines. But as always, the devil is in the details. Here are some specific figures that will make your head spin. Within the dominant group R1b, there is a subclade that plays a key role in our story, R1b-M405. It encompasses several subbranches and constitutes 34.15% of all male lines in the Netherlands. Over a third of Dutch men carry this marker. But the most interesting part is its geography. Spatial analysis revealed a pronounced trend. The frequency of R1bM405 increases from south to north, and its key subbranch R1bU106 in Brabant, on the border of the Netherlands and Belgium, demonstrates a clear gradient, 37% in the north and 21% in the south. The highest frequencies of these lines are concentrated in the Netherlands, northern Germany, Denmark, and southern Sweden. This area is what geneticists refer to as the Northwest German Array. Remember this name. It will appear in our investigation again when we reach ancient burials in England. The second most frequent male line in the Netherlands is haplogroup IM170. Almost every 4th Dutch man carries this ancient marker. In Europe, it has 2 major centers. The first is Scandinavia, where the subclade I1M253 predominates. The second is the Balkans, with the Dinaric subclade I2. In the Netherlands, spatial analysis revealed a clear trend. The frequency of IM170 increases from southwest to northeast, that is, towards the North Sea, towards Friesland. Notice the coincidence. R1B-M405 increases from south to north. I-M170 increases from southwest to northeast. Both trends converge at a single point, on the northern coast, where the Frisians historically lived. It is this combination of a high proportion of R1BU106 and I1M253 that creates this people's unique genetic autograph on the male line. An autograph that unites them with the populations of the entire North Sea coast, from Denmark to Lower Saxony. And with someone else. But more on that later. But here's what truly boggles the mind. Male and female genetic lines tell completely different stories. According to mitochondrial DNA, which is passed down from mother to children, no significant spatial structure is found in the Netherlands. Maternal lines are evenly distributed throughout the country. No gradients, no clusters. But as we have already seen, the picture is quite different for the Y-chromosome. Pronounced trends, local peaks, clear boundaries between the north and south. Scientists explain this paradox with the patrilocality of traditional societies. Men remained on their ancestral lands generation after generation. Women moved to their husbands' families, gradually mixing maternal lines throughout the country. It turns out that male genes have preserved a geographical memory of ancient tribes for centuries, while female genes have long erased it. This is why the Y-chromosome has become the main key to unraveling the mystery of the Frisians. And this key will now open an astonishing door for us. Now, let's go back to 2002, to the study that has become a classic of population genetics. Weale and co-authors analyzed the Y-chromosome in 313 men from 7 British towns, arranged in a line from East Anglia to North Wales. They discovered a striking contrast. 5 towns in central England were extremely homogeneous in terms of male lines. But 2 towns in North Wales were sharply different from them. In Wales, the Atlantic branch R1B-P312-L21 predominated, characteristic of the ancient pre-Anglo-Saxon population of Britain. Meanwhile, the R1B-U106 line, the very marker of North Sea Germanic peoples, was nearly absent there. Wales turned out to be a genetic fortress where Anglo-Saxon male lines never penetrated. The mountains became a barrier not only for armies, but also for genes. Central England and Wales are one country, but 2 different genetic worlds. The researchers then asked, if the English do not resemble the Welsh, then who do they resemble? The answer was stunning. When the researchers compared the data from central England with continental samples, they found that the English and Frisians showed nearly identical spectra of Y-chromosomal lines. All comparisons confirmed the absence of significant differentiation. At the same time, central England was noticeably different from Norway and sharply different from Wales. Researchers then attempted to explain this coincidence without a mass migration. Perhaps it was just a continuous exchange of people across the North Sea over thousands of years? The model showed that such an explanation would require a continuous flow of about 0.3% of men per generation. No demographer would confirm such figures. However, if mass resettlement occurred about 1,500 years ago, everything falls into place. The best model required that 50 to 100% of the male gene pool of central England during that period derived from continental migrants with a genetic profile similar to that of the Frisians. But can this model be confirmed directly? Now let's look at the data obtained by a completely different method. Up until now, we've been talking about the Y-chromosome, father's lines. But in 2016, Schiffel's team went further. Complete sequencing of the genomes of 10 ancient individuals from East Anglia. Their remains spanned the period from the Iron Age to the early Anglo-Saxon period. And here is what was discovered. Anglo-Saxon samples from the 5th to 8th centuries were found to be closest to modern Dutch and Danish individuals based on rare alleles. Iron Age samples, that is, before the arrival of the Anglo-Saxons, were closest to the Irish and French. The contrast is striking. But the most important thing, according to autosomal data, the modern population of East Anglia carries on average about 38% of the Anglo-Saxon component, with a range from 25 to 50%. This is no longer just male lines. This is the whole genome. And it points in the same direction, towards the North Sea coast. Here's a story that will give you goosebumps. Remember the study by Gretzinger we mentioned earlier? 460 ancient genomes, the largest sample from the Anglo-Saxon era. Well, the results shocked even the authors themselves. In early medieval burials from East Anglia, the average share of the continental Northern European component reached 76%. 3/4 of the genome came from the continent. But an even more dramatic picture emerges for the Y-chromosome. Almost complete replacement of male lines occurred. Instead of the hallmark of Bronze Age Britain, R1b-P312-L21, other haplogroups began to dominate in the burials. R1b-U106, R1a-M420, I1-M253, I2A1-L460. Recognize the set? These are exactly the lines currently dominant among Northern Germans and Dutch. The Frisians are part of this genetic world, not mere observers but key actors. So, what have we learned when we compare all the data? The best genetic source of the Anglo-Saxon component is the strip along the North Sea, from Friesland through Lower Saxony to southern Sweden. There is particular similarity with early medieval samples from northern Germany. Now pay attention to the contrast within the Netherlands itself. The southern European branch R1b-S116, also known as P312, comprises just over 21% and increases toward the south of the country. This line is more characteristic of France and Iberia. But northern Netherlands is a completely different genetic world. There, R1b-U106 and I1M253 dominate, the same set we have seen in Anglo-Saxon burials. A clear picture emerges. Within one small country, an invisible genetic boundary runs. To the south of it, continental Europe. To the north, the North Sea world. And it is this northern autograph that swam across the channel 1,500 years ago, laying the foundation for the English gene pool. Now think about this. Modern English people are no longer identical to those early medieval settlers. Their gene pool has been diluted with Celtic substratum, Norman influence, and later migrations. According to rough estimates, the Anglo-Saxon share in southeast England amounts to about 25 to 40%, and in other regions, it is even less. England has absorbed too many layers. Meanwhile, the Frisians remained in place. Yes, they too did not live in a vacuum. An influx of southern Dutch lines, Scandinavian influence in the early Middle Ages. But among all continental Europeans, it is the Frisian North German-Danish belt that best preserves the genetic profile from which the early English gene pool once branched off. By the way, the line R1a-M420 constitutes only about 4% in the Netherlands. It is associated with the Central and Eastern European component and emphasizes how North Sea, the Dutch genetic landscape, remains. And now about what I promised at the beginning. Ancient DNA data showed that the Anglo-Saxon migration was not a raid of warrior bands. The continental Northern European component was carried by both men and women. The gender imbalance is minimal. Entire families made the crossing across the sea. This fundamentally differs from, say, Viking expeditions where men predominated. The scale of the resettlement turned out to be enormous. But what about the other side? The Dutch gene pool remains surprisingly stable. Even minor lines, haplogroup G at about 2.7%, haplogroup J at 3.45%, have maintained positions similar to those of centuries ago. Does this mean that the Frisians are frozen Anglo-Saxons? No, they also changed. But among all currently living continental populations, they remain the closest genetic portrait of the people who crossed the North Sea 1,500 years ago and laid the groundwork for what we today call England. What do you think? Do these genetic discoveries change your perception of the origins of European peoples? Two shores of one sea, two languages, two countries. But underneath all this, the same genetic foundation laid 1,500 years ago. Did you know that genetic twins can live on different sides of the channel, speak different languages, and not even suspect how close they are? If you live in England or the Netherlands, you surely have something to add to this investigation. Maybe you have done a DNA test yourself and found unexpected matches? Write in the comments what genetic mysteries of peoples you would be interested in exploring in future episodes. And be sure to subscribe to the channel so you don't miss new investigations. There are still many stories ahead where ancient DNA overturns everything we considered an unshakable truth. And lastly, do you know what unites the word freedom in Frisian and Old English? It sounds almost identical. Frisian, freedom, and Old English, friodom. Coincidence? Hardly. Medieval Frisians were renowned for their fierce love of freedom. They knew no feudal system longer than any other people in Europe. Their land was called Frisian freedom, a territory where there were neither counts nor serfs, and decisions were made at popular assemblies. And what's intriguing is that the same model of local self-governance laid the groundwork for Anglo-Saxon witan, councils of the wise. Perhaps not only genes and language ventured across the North Sea 15 centuries ago. Perhaps the famous English spirit of freedom, which later found expression in the Magna Carta and parliamentary tradition, was born not on the British Isles, but on the flat, windy shores of Friesland, and still lives on both sides of the sea, recorded not only in chronicles, but also in DNA.